Okay, that's okay. You do that part. Thank you. So it's actually broadcasting now, right? Okay. Oh, that sucks. Okay, if you don't mind, I'm going to get going. I won't try to keep you too long. I know some of you have travel plans because of reading week break. I'll try to take you through things as quickly as I can related to the lecture material and what's going to happen next week in terms of your midterm as well. So some of you, before you actually showed up here, took part in a small experiment where I asked you to pick whatever hot beverage that you wanted to have. Some of you chose tea, some of you chose coffee. Um, for those of you who actually did that, which one did you pick, coffee or tea? Tea? Why did you pick tea? You already had a coffee this morning. Excellent answer. Great. How many of you, if I asked you, out of these three choices, whether you have green tea, black tea, or coffee, how many of you believe that green tea is the healthiest option out of those three? Okay. How many of you think that coffee is the healthiest option out of those three? And black tea? Not a few hands going up for black tea. But most of you sort of put your hands up for the green tea option. And I want to actually come back and talk to you a little bit about that a little bit later on in the course. So we're going to finish off this section that we have right now on colorectal cancer. I'll finish off with a couple of odds and ends related to the course, and then I'll start telling you a little bit about the end of the course as we go on past the midterm next week. And yes, we are still going to do a little Twix experiment to see if Twix actually does improve your cognition, or if you actually believe that Twix actually makes you at all smarter, remember better, or anything else. We're talking right now about colorectal cancer. And just the other day, on the news, we had an individual who is now going to devote his life to colorectal cancer. And why is he going to devote his life to colorectal cancer? So you have to keep a couple of things in mind. This is an individual who is part of Google, and as part of Google, he has access, and as a senior administrator in Google, he's paid well. He's in one of the best areas in the country with great healthcare, better healthcare than we have here because he can afford that healthcare in the United States. And his wife still passed away from colorectal cancer. Now, Google is also very interested in the biology of cancer, as well as getting technical expertise in cancer research as well. And whether you believe so or not, with the University of Toronto, we have a number of collaborations where we are working with Google to manage large data sets. And Google is very interested in your health data management associated with that, including genotyping, understanding the genes, etc. And so colorectal cancer doesn't really care if you have a lot of money and if you have lots of screening done or not. If you're not getting screened and it's too late, it doesn't matter if you have $1 million or if you have $10. It's a lethal disorder. And again, you have to keep in mind that he is part of the 1% in America who could afford the best health care for his wife. And yet now his wife has passed away, even with this vast array of resources that are available. So I'm going to take you through and revisit this colorectal cancer module as we finish it off. I'll take you through some of the ideas that have been brought up in the literature as well as in research related to colorectal cancer and why we think colorectal cancer might be diet related. And then we'll sort of segue into what you should expect in the rest of the course. And the one thing that I mentioned to you, yes, we're going to ask you to learn lots of different things in this course that you are going to have to memorize, and that's the cognitive things that we want you to take away. Like, we learned about all these different pathways, we learned about all these molecules, but then there's also the affective things that you will take away beyond this. And when you hear, for example, that red meat causes colorectal cancer, do you believe that right off the top when you see that the World Health Organization has just said that red meat is now a carcinogen, or processed meat is now a class 1A carcinogen? I want you to be really critical thinkers. Yes, I want you to memorize things that I think is important so that you can have a foundation, but I also want you beyond this course, after this course is over, as you get into third year, as you go off into professional schools or into research, to become really critical thinkers. And I'll be setting you up a little bit about what I'm going to try to do to get you ready for that in the next sets of modules. Again, when we look at colorectal cancer risk, we know that there are things that are related to the large intestine or the colon that is really important. And if you have certain types of inflammatory bowel disease, and please don't memorize the numbers that are associated with this, but we know that there is a fairly large risk if you have inflammatory bowel disease. And the key part here is not that it is in the bowel. The key part here is that it is inflammatory related. Or if you have a genetic predisposition to having issues in the colon, that there is a risk associated with this as well. But even without that, as you get older, you have a general risk that increases every year that you get a little bit older as you have your base lifestyle. And this 5% population is actually growing. Every single year that you get 1 year older, that 5% creeps up, and it will continue to creep up as you get older, as the general population gets older as well. Now, we don't know a lot about colorectal cancer. We only have some ideas about colorectal cancer, which is what this lecture is really about. So this is a busy slide, and there's a lot on this slide, and I put a lot of text on the right-hand side of your slide in case you miss writing down some of the details. In the past module that we've been looking at red meat, and we've tried to identify red meat, and we wanted to understand how red meat might be implicated in this. The World Health Organization says that red meat causes colorectal cancer, or is strongly linked to colorectal cancer. How do they come up with these different types of proclamations, and what's the mechanism? For us as biologists sitting here at U of T, we want to know, how does eating red meat actually produce the types of things that they all say? Well, we know that in red meat, a lot of things are metabolized to this first compound, nitrogen oxide. And this nitrogen oxide that you form in the early part of the digestive system, as you swallow it and as it goes down into toward the stomach, this will actually produce those N-nitroso compounds that you've been hearing about in your tutorial, as well as what we've been talking about in the last few lectures. So this itself is not so great for you, but we also know that in red meat in particular, we have another substance known as heme, and heme is one of the other factors. The heme iron and different versions of the heme iron can help to catalyze and speed up more of this N-nitroso compound formation. And so we know that when we have all of these different types of factors in your gut being exposed at the same time, that it might speed up the process of tumor formation in the colon. We also know that heme iron, and this will become part of today's discussion as I go through a PNAS paper with you, can also act on polyunsaturated fats that you will also take in your diet, and it will react with them in the gut to produce reactive oxygen species. And we know that reactive oxygen species, especially in the gut, can have a number of deleterious effects on your gut lining. And we're going to come back and examine this a little bit later on. So all of these things together, so taking in red meat, which has all of the nitrates and nitrites that lead to the production of the N-nitroso compounds, which then get further amplified through other aspects of the digestive tract or the other parts of the diet that you're taking in, will all impact your colon negatively. But again, keep all of this in mind. We're talking about the colon. We're talking about red meat. We're talking about other factors. And this is one potential mechanism that I'm introducing to you. We know that gut inflammation as well as your diet, directly or indirectly, can also heavily impact what happens in your gut as well. So in the past, and I'll show you a diagram of why we think that this is important, colorectal cancer has had a strong association. It's not a definitive cause, but a strong association with diets that are higher in red meat. Keep in mind that most of the different things that you read about will also indicate that white meat, as well as fish, don't have this association with colorectal cancer. And I'll come back to this as we wrap up this module. Keep all of this in mind as we go through today's talk. I told you that heme, which has that iron component and that porphyrin part of that heme molecule, this is one of those things that helps to exacerbate, accelerate, amplify all the bad things that we find in our diet. And again, heme, why is one of the things that is bad with red meat? We find it at much higher levels in red meat overall. So why do we think that all of this is bad? If you look at a number of different studies that have been published, and you don't have to worry about trying to memorize this. Obviously, it's not something that I want you to concentrate on too much. But if you take a look, all of the studies that you see implicated here indicate that if you take in red meat, that you are at an increased risk of developing colorectal cancer. And this has been a common finding over the last 20 years, that red meat has a strong link, a strong association with the formation of colorectal cancer. So it's not one scientist that's finding it. Many labs, many scientists have been repeating these experiments over a number of years and finding exactly the same thing. The biology is sound. Red meat seems to cause an increase in colorectal cancer. So we want to understand the biology, because if we understand the biology, maybe there's an intervention that we can initiate to stop this colorectal cancer. And this goes back to that heme. That heme is that central player in all of this. Red meat has more heme. Heme is amplifying, exacerbating, and making the situation much worse. And heme has a number of things that are going against sort of the health status of your colon. And we know that heme can directly cause the proliferation in some models of the mouse mucosal lining through that lipid oxidation. And I just mentioned to you in the previous diagram about lipid oxidation, reactive oxygen species. So heme itself can directly do this. It can also be converted into another form known as hemin, and this protoporphyrin 9 iron oxide compound can also cause deleterious effects within the digestive tract as well. We don't absorb this hemin extremely well, so it stays in the digestive tract all the way down into the colon, and then we can also excrete it. But again, we don't see a lot of cancer in the stomach. We see a lot of it in the colon. And again, that was one of the things that we had in our last lecture. So this hemin molecule is also very catalytic and can stimulate those reactive oxygen species that we looked at before. And if you also have those polyunsaturated fats, then it may be even worse. So all of these things suggest that heme is sort of the toxic compound. If you directly give rodents heme, that heme molecule, the unbound iron, and it gets converted into that protoporphyrin, the contents within the colon also become more cytotoxic. So any food that they have there will become cytotoxic to the mucosal lining. And that cytotoxicity can also directly injure the rodent epithelial surface cells. So if your cells get damaged through cytotoxicity, it can actually cause this hyperproliferation. This hyperproliferation is what causes those polyps, those adenomas to form, and if unchecked and untreated, will eventually cause that tumor growth within the mucosal wall of the colon that we looked at in the last lecture. So with the inhibition of apoptosis that unfortunately we're not going to be able to cover, this overproliferation or hyperproliferation eventually will lead to a classic sign in cancer, which is hyperplasia. And this is that adenoma and the colorectal cancer. So heme itself seems to be, in a number of studies, bad. So red meat is bad. Red meat is bad because of heme. Heme directly causes cytotoxicity through reactive oxygen species, hyperproliferation, etc. And we know that overall, we don't really absorb heme very easily. So when you take it in through the diet, many things get absorbed at the level of the small intestine. For those of you who have had a physiology course, you'll know that in the digestive tract, when we take in food, the small intestine is really the site where we get the biggest amount of absorption. So you take in food and you absorb the breakdown products at the level of the small intestine. Heme is not one of them. Heme stays in your digestive tract, and it stays in your digestive tract all the way through to the colon. And then again, even there, we don't absorb it well. It just stays in the colon, produces cytotoxic compounds, and then gets eliminated in your feces. So we know that there might be an effect through the microbiome or the microbiota that lives within the colon. And we know that if you have a high red meat diet, that in the feces that are produced after a red meat diet, that you will also have an increase in the number of bacteroids that get excreted as well. And we know that a heme-rich diet will also directly impact the microbiota that lives in your gut, increasing the overall population of the gram-negative bacteria. And I'll come back to bacteria at the end of today's lecture again, just to give you a little bit of a heads up on what to expect on the midterm. Do you have any questions about this before we go on? No? So another factor that I wanted to introduce just before going on is why else is heme really bad? So the gut microbiota, in theory, can produce hyperproliferation and can do so in ways indirectly through modulating oxidative as well as cytotoxic stress. The oxidation that is a result of the different bacteria that are in your large intestine can actually peroxidize lipids, and this can also cause the formation of what is known as cytotoxic heme factor. So having heme produces cytotoxic factors. These cytotoxic factors induce lipid peroxidation, and it's a positive feedback loop. Those lipid peroxides can also directly act now on the remaining heme that we don't absorb really well, that's stuck in the large intestine, to also produce this cytotoxic CHF factor. So it's a very vicious positive feedback loop that also increases even more so the cytotoxic stress that is already on the large intestine. So again, just keep in mind that we're looking at the large intestine, and if you're thinking about this, why doesn't it have this big effect on the small intestine? We know, for example, that if you give mice heme in their diet, that where we see all the issues are here in the large intestine. The small intestine, even though it's long and the heme stays there for long periods of time, we don't see small intestinal cancers. We only see it in the colorectal tract. So you might be thinking, why is that the case? You know, the small intestine is actually much longer. The heme is there for much longer periods of time. But why is it the case that we have more of this issue at the level of the colon? And it probably has a lot to do with the gut microbiome. And this is sort of the idea that these individuals wanted to pursue as part of the PNAS paper. They wanted to know what makes this difference. So why do we see colorectal cancer in the colon? You know, heme is there. Heme, in theory, can interact with a number of things. We know that there's a difference in the population of the different bacteria in the small intestine versus the large intestine. Is it one factor versus the other that tends to dominate in producing this colorectal cancer? Is it really the gut microbiome, or is it the red meat itself, and there's something different about the epithelial cells? What accounts for having large intestinal cancer versus small intestinal cancer, which we don't really see? So their main ideas. What is it really heme-induced? Does it involve the microbiota at all? How is this related to hyperproliferation? And do they play a role in this cytotoxicity using the mechanisms that I've just introduced to you? So I'm going to take you through the data really quickly. And there are a couple of graphs and there are a couple of figures that are coming up. I want you to feel comfortable reading and understanding the different types of primary literature that we introduce for you. And the experiment itself is really, really easy. If I think that the gut microbiome, the gut microbiota, all of the bacteria that live in my gut is responsible for producing these cytotoxic effects, why don't I just get rid of them after using antibiotics? Because we know antibiotics will do this. So these mice that they were using in these studies were divided into 4 different groups. There was a controlled diet group. There was a heme diet group. They had a supplement of heme introduced into their diet, so it should produce cytotoxic changes, as I've just showed you earlier. A controlled diet where we wipe out the gut microbiome using antibiotic treatment. And oftentimes, if you see X, it means treatment, so prescription or treatment, etc. So ABX representing antibiotic treatment, where they have the control diet, but all the gut microbiota have been wiped out. And then the heme diet with antibiotic treatment as well. And they wanted to know, after 2 weeks of this, given a diet either having lots of heme, with and without the gut microbiome, what is the end result of all of this? So one of the interesting things that they found is that just giving heme led to a lower body weight. And again, they go through some explanations, and I'm just introducing this because there's a table coming up soon on this. And overall, the fecal dry as well as wet weight was increased a lot in the heme group that was treated with antibiotics. Gram-negative, the bacteroidetes, as well as the firmicutes with this antibiotic treatment. Didn't matter the diet. So whatever you were doing, if you gave them antibiotics, whether you were the control diet or whether you were doing the heme diet, you wiped out bacteria. Okay, so there was no protective effect of diet on this. If you give them antibiotics, the total bacterial counts go down. That's shown on the left. And then all of the different types of bacteria, whether it's bacteroidetes shown here or the firmicutes shown here, you wipe out both gram-negative and gram-positive bacteria after you use antibiotics. And again, the details of using ampicillin, neomycin, etc. are not so important here. So the antibiotics are effective. They're wiping out these different bacteria, and they're wiping out both types, gram-negative as well as gram-positive bacteria. We're wiping out the ones that are beneficial, the firmicutes, and we're wiping out the bad ones, the bacteroidetes as well. So if these bacteria are really that important in toxicity, we should start to see an effect when we go back and we start looking at them. So one of the things that we have implicated these bacteria in doing is that they might play a role in lipid peroxidation, so breaking down and creating reactive oxygen species. So how do you measure lipid peroxidation? So this is the one thing I want you to take away from this slide. If you're measuring lipid peroxidation, we look for the presence of a chemical thiobarbituric acid, reactive substances, or T-bars. If it goes up or down, it tells us about the lipid peroxidation state. And we can measure how much of this T-bars is actually being produced in the waste that is coming out of the animal. And it turns out that the T-bars, that is a measure of this lipid peroxidation, was actually low in the control and the control antibiotic groups, and increased in the heme groups. So this meant that heme was actually causing lipid peroxidation. Was that surprising? Probably not. The fact that heme causes lipid peroxidation has been known for a while. And if you have heme with antibiotics or without antibiotics, the T-bars level were really high. There was lots of lipid peroxidation. But remember, antibiotic treatment has dramatically decreased the levels of our gut microbiome. So if we're going back to our hypothesis that it's maybe mediated through this gut microbiome or the gut microbiota, and we've wiped out our gut microbiota, both the bacteroidetes and the firmicutes, and we still see an increase in the overall levels of the reactive oxygen species, the T-bars that represents lipid peroxidation that we think might be bad for the gut, this likely suggests that the bacteria don't play a major role. It's unlikely that they play a major role in the overall lipid peroxidation state. So it suggests that it's really the heme, that it has nothing to do with the types of bacteria that might be involved. And again, it's mainly for your own information. Please don't try to memorize these results. If this is a measure of lipid peroxidation, and as a result, the reactive oxygen species in a controlled diet, these are the basal amounts, and we see that between the control and the controls that are treated with antibiotics, so all the gut microbiota have been wiped out, at least the bacteria, both gram-negative and gram-positive, we don't see a huge change in the lipid peroxidation state. So if we then go and take a look at the heme molecules, the heme-treated, the ones that have more heme, kind of representing the red meat diet, when we go and take a look at what the effect of heme is on them, and we look at their lipid peroxidation state, what we see is that the T-bars that represent an increase in lipid peroxidation and therefore likely an increase in reactive oxygen species actually dramatically goes up after heme. Again, not surprising. But it stays high even if we wipe out the gut microbiome. And so, again, it suggests that it is not mediated primarily through the gut microbiota. It's mediated through the heme and its cytotoxic effects directly. And in terms of cytotoxicity, you see the exact same trend in the bottom line. So they wanted to know, so if heme is really doing all this stuff, what sorts of things does it actually account for? Do they see the same types of results that I've indicated for you a little bit earlier on? So this is a slightly busy slide, but I want you to understand this slide because it's really important. You're biology students, and so learning a little bit about histology and a little bit about immunohistochemistry. So histology is in these 4 panels over here, and immunohistochemistry are on the 4 panels to the right. If we were to take a look at what we see with normal histology on what we call an H&E stain, a hematoxylin and eosine stain, which gives us this characteristic pink and purple stain that you see in all those histology textbooks, it tells us a lot about structure. So even by naked eye, just taking a look at it, if you look at the control diet after 2 weeks and you look at the colon, and you look at what happens after giving these animals heme diets for 2 weeks, and this is taken at the same scale, there's no magnification changes or anything else, one of the things that you can see very clearly is in purple up here. I'm hoping that you see this in purple at the top. So these represent the sort of villi and the crypts. So these are the villi, your fingers would be the villi, and in between your fingers would represent the crypts. And what you see very clearly is that after just 2 weeks on this diet, what is the relative size? Which one is bigger? The one on the top or the one on the bottom in terms of the size of the purple staining that you see? So the ones on the bottom here, you can see that these are much, much longer. It's like your fingers have like grown much longer. They have actually proliferated. They have grown much longer here. Now, again, this is just on heme. So on heme, we know that we have more cytotoxicity. We know we have more lipid peroxidation. But these overall crypts, the villi, the depth of these crypts has actually grown. They've actually started to proliferate. Proliferation is not a good thing for us to have within the colon. So if we go back in and we take a look at what happens after antibiotic treatment, we don't really see much change in the control condition. So this is control with antibiotics now. And in the heme condition, it's still bigger, but the numbers have gone down. So what accounts for this proliferation that we see histologically over here on the left? If we then go back and we take a look at the same sections, so the same treatments, control here without antibiotics, control animals, control diets with antibiotics here on the right, and down here the heme diet, one of the things that we can pick up on an immunohistochemical stain where we are adding an antibody to a marker, and one of the things that we want to mark is proliferation. We can identify a marker protein in proliferating or dividing cells known as Ki67. So if you have a cell that's dividing or a proliferating cell, we would use an antibody to Ki67 to pick it up. And that's one thing that I would like you to make sure that you take away from this slide, that we can actually identify proliferating cells using Ki67. So Ki67, all of the stuff down here on the bottom, the dark brownish stains that you see, this is where we have Ki67 positive cells. And these Ki67 positive cells, we see lots of them. Is that surprising? Where are our stem cells within the large intestine? They're in the crypts, and so right at the base of where your fingers are, the villi, in the crypts, we see lots and lots of this brown staining, which you see over here. So this would be one crypt, this is another right beside it, and the little brown spots that you see at the bottom represent the Ki67 proliferating cells. But if we take a look at the heme, we actually see more of them. So these are certainly bigger, longer, as we saw over here. But if we take a look, we see also more of these dividing or proliferating Ki67 positive brown spots down here. The antibiotics tends to wipe them out more or less. And if we were to quantitate them, we would see that with heme, we see more of these Ki67 cells. The total number of cells is also increasing. And heme is causing this proliferation of these cells that we can mark through Ki67. So heme itself is directly causing the increases that we see in these dividing cells. It is causing the proliferation. Do you have any questions about this slide or about Ki67 or about the histopathology that you see here? Yes? Sorry, which? Which of the panels is Ki67 belong to? So if you were to take a look, sorry, let me just get the laser pointer back. Which ones are they positive? So every panel here is positive for Ki67. So this little C shape down here, that's the crypt. Every single cell in that crypt is Ki67 positive. So you have multiple cells down here. But if you take a look, the number of brown stained cells which are Ki67 positive is much bigger down here in this panel. Every one of these panels actually shows Ki67 positive cells. Any other questions before we go on? So this is a really complex model. I don't need you to memorize it, but what it suggests is, and please don't try to memorize it, this is just a take-home message for you. The whole biology of why red meats might cause cancer in an animal model is probably a whole host of things. For sure, and this paper also confirms previous findings, that there is something that involves this heme molecule, and that heme molecule probably does produce lipid peroxidation. It probably produces those cytotoxic heme factors, and this may increase the level of reactive oxygen species. They definitely showed that. But it's probably much more complex than that. They know that from the antibiotic studies that the gut microbiome helps to increase that activity. And whether or not red meat does it in this particular model that is being shown from their paper, where some of those cytotoxic compounds, the reactive oxygen species, is directly causing damage to the surface epithelium, is not really clear in this paper. And we don't really have a mechanism that accounts for everything that we know about red meat, heme, as well as colorectal cancer. And again, you have to keep all of this in mind. This is not a human study. This is an animal model study where we think we know a lot about the mechanisms and they develop tumors, but it may not exactly mirror what we see in humans. Because based on these mouse model studies, this is a diagram that I'd like you to be familiar with, and I'm going to tell you what you should be familiar with. I know most of you are thinking about the midterm, but I want you to circle this. Come back to this for your final exam. We know that in the normal epithelium, where there's no damage going on and you're not eating a lot of red meat, where you don't have a lot of lipid peroxidation, everything is fine. Some of you will be able to go through your entire life without any worries whatsoever, even if you eat red meat. However, we know that in some individuals, and this is a genetic factor, that if you lose the gene APC, which is a tumor suppressor gene, that this can actually, in the right instances where we have an increase in reactive oxygen species at the level of the epithelium, that they might start to proliferate. You'll start to form those little polyps. You'll start to form those adenomas. You'll start to develop those early adenomas. And again, even there, if you remove those adenomas, there's no issue. However, we also know that some individuals are genetically disposed or predisposed to developing full-blown colon cancer even after you've removed those polyps. And we know that there are a number of factors that also come into play. So not only do you have to lose a tumor suppressor gene like APC, and by the way, there is a lot of evidence to suggest that these are de novo mutations or de novo losses of function. So you don't have to have anyone in your family that has had any issues with APC. You could be sitting here and something could go wrong with your APC, and your APC tumor suppressor gene may suddenly decide that it's not going to function as well as it once did before. And these sporadic forms of APC mutations are well documented, and they have been shown for over 50% of losses of APC are sporadic. They appear out of nowhere. But we also know that in order for you to get a full-blown carcinoma down here, that a number of other factors have to be in your genetic background. You also have to activate KRAS pathways that we'll see in other systems in the rest of the course. If you have a deletion of certain genes on the Q arm of the 18th chromosome, that will also be effective. And if you lose the P53 tumor suppressor gene, that you're more likely to develop full-blown cancer. So we know that there are a whole host of things. It's not just red meat that's important. You also need some genetics. You also need some tumor suppressors being turned off. And we don't understand all of those mechanisms that come into play in this particular model. The one thing that I'm going to ask is that you do go back and check out, for example, the different factors that are involved in tumor progression. So you should know, for example, that the loss of APC might cause hyperproliferation, that after you form an early adenoma, that KRAS activation in epithelial cells might lead to a more aggressive adenoma, that certain factors that are lost on the 18th chromosome might lead to a really progressive form of an adenoma that leads to a carcinoma. And we know these from mouse studies. We know these from human studies when we do pathology on them, that all of these factors come into play. So again, keep in mind a couple of things as we sort of wrap this module up. We know that red meat causes lots and lots of damage. It causes the generation of mutagens, the type of cooking that is important in previous lectures that we've gone to, may lead to polycyclic aromatic hydrocarbons, etc. And that is important, and that's an important thing to keep in mind. But keep in mind this as well. I mentioned at the very beginning, it's been associated with red meat. And grilling red meat, cooking red meat at high temperatures has been associated with increases in certain forms of colorectal cancer. But we grill a lot of other meats, and we still form polycyclic aromatic hydrocarbons, PAHs, after grilling chicken. We still form this after grilling fish, but we don't see cancer associated with grilling. So there's a lot of complexity there. Why is it worse when we grill red meat? Is it only the heme that accounts for it? Is it only the microbiome that's interacting somehow? We have no answers for that. And in fact, it's a very complex type of question. We don't know what the causative link really is. It's something that some of you might want to investigate. Why is grilling some forms of meat, when we know that carcinogens are produced by any type of grilling on any meat, so bad for us in terms of colorectal cancer? The other thing that one of you had actually asked, and it was a brilliant question, I thought, how come animals don't get cancer? Because we don't really see colorectal cancer in animals. Other animals eat red meat. Why don't we see, if heme is so bad, and if heme is something that is so bad in our digestive system, why don't we see lots and lots of colorectal cancer in other animals that eat red meat? And we don't. It's absolutely true. When you go through the literature, in animals, generally speaking, we don't really see a lot of them having colon cancer. So why not in other carnivores? And it's a great question. I don't have a good answer for you, but that's one of the things that I'm hoping that you take away from this course. That we don't always have the answers, and some of those answers might be ones that you want to investigate. So this is sort of the end of the module that we're looking at related to the gut microbiome and some of the pathophysiology associated with the gut microbiome. We're going to start looking at different aspects of what we talked about, though. So it's a continuation in some ways. You choose to give yourself cancer every time you eat red meat, whether or not you know the mechanisms. Right now it's observational, but it's a strong observational link to red meat and cancer. We also know that you can develop cancer if you're physically inactive, and you also choose to be physically inactive. How many of you have heard that exercise is good for you? All of you, right? But why is exercise good for you? Have you ever given that any thought? And in the next few modules, we're going to look at whether or not exercise is really all that good for you. And if there is a biology associated with it. You've heard that red meat causes cancer. We've looked at the biology. Do you know the biology behind exercise? Why does exercise make your brain better? I'm hoping that by the end of that, you'll actually understand that. And this gets into the biggest controversies right now in science are actually in the whole field of exercise biology, in terms of, is exercise actually good for you? Do any of you know any factors that have been published related to exercise? No? Yes, at the back.
There's evidence that it has across the board, like effects on decreasing just like morbidity.
Okay, so it decreases morbidity. Absolutely true. How does it decrease morbidity? It's like magic. We don't really deal with magic here. How does it do it? So right now, let me ask you a slightly different question. So if you read a paper that came out of the University of Arkansas, would you believe that that research is just as good as the research coming out of Harvard University? Harvard University. Yes? So two of the biggest researchers right now, Bruce Spiegelman as well as Amy Wagers, have suggested in the last 3 to 5 years that we understand why exercise is good for you. So we're going to learn about these molecules. But they're also under the greatest constraints because not everyone believes that they're right. But they're out of Harvard, right? So Do you believe everything that you are going to read in the papers that you read? And how are you going to differentiate if someone tells you magically out of the blue, and this is where that Twix comes in, um, how do you know if someone tells you that Twix is good for you that you actually believe that research? Sorry? That's a good answer. So I have some Twix for you. Please help yourself at the end. Twix is actually probably not so good for you, to be honest. Chocolate, if many of you have actually seen, chocolate is good for you, chocolate is bad for you. Coffee is good for you, coffee is bad for you. How do you make sense of all of that stuff? I want you at the end of that module to also realize that in science, we have a lot of controversies in science related to GDF 11, related to PGC1 alpha, related to Irisen, all the stuff that is published in Science, Nature, and Cell, we don't know if it's actually right. And that leads to the next module on bioethics. So as research scientists, how is our research changing? Every scientist in the next 5 years will be barcoded. You will have your unique barcode so that we can track your research. Why did we get to that stage? Because our science is so faulty. It's so irreproducible at this point that it's actually a big issue right now in science, whether or not we have a problem with being able to reproduce data that's published in some really high impact journals. From there, we're going to go into things related to other aspects of bioethics. If you can wipe out a whole species of animals, should you go ahead and do it? Because right now, that's the debate that's going on in Brazil. Should we wipe out all the mosquitoes that carry the Zika virus? Should we actually pay for this? And how should we go about doing this? Do we just wipe out all mosquitoes? Or should we try to be more, I guess, judicious about this whole process, especially in light of the fact that we know nothing about the biology of Zika. And this gets into the technology. So many of you have heard of CRISPR-Cas9, and I want to take you through the biology and the molecular biology behind CRISPR-Cas9 and what its current limitations are and where we think it's going to go. And ultimately, at the very end of the course, we're going to talk about precision medicine and how once we think we can modify your genome, how that might impact you in the next 10 years. Because precision medicine now is at the point where it can impact you if you go to the hospital down the street. So this is where we're kind of heading. And these are the types of things that I want you to think about. We have an extra tutorial. By the way, if you're worried about your midterm, please don't. It's a very fair midterm. I've actually tested it out on a couple of different individuals, and they all failed, but that's okay. They're not actually in HMB204, so I kind of expected them to fail. But I also asked a couple of other individuals who knew a little bit about the course to take it, and they did pretty well on it. So I think it's a fair midterm. A couple of things. If you're downtown on campus, we're going to have an extra review session after Family Day. So drop in. It'll also be streamed for you. If you have questions, please post them however you want, whether it's on the discussion board or through social media, and we'll try to answer that for you. On Monday the 22nd, the lecture is going to be devoted to a full review to make sure that you're prepared. You still have a few days to get ready. In the worst case scenario that you're not ready by Monday, you can still get ready by Friday, okay? So a couple of other things. It's just to lectures 1 to 8. None of the tutorial material at all will be tested. I like asking some more challenging questions. What I like to do is actually go through the bullet points to see if you've actually read the bullet points as I presented them to you in class. And I usually take my multiple choice test questions directly from them. I don't take things off of figures that I haven't really covered well. Okay, so do you want an example of a question? Of course you do. You just aren't saying that you do. Do you actually want the question? Yes. So this is the question. The answer choices, this is exactly as how it's going to appear on your midterm. Exactly like this. Now, if you're thinking about this is actually not an easy question to answer because you've actually heard about lactobacilli in about 2 to 3 different lectures. So you're going to have to go back when you're studying for lactobacilli. You're actually going to have to try to figure out, wow, you know, lactobacilli, we've heard about it in lectures about the skin, we've heard about the colonization of the gut microbiome. We've actually heard about it in one other lecture as well. So there's a whole bunch of things that you actually have to be on the lookout. But this is an example of an integrative question. You'll get this question, and you'll get some things that are below it that if you studied up on lactobacilli. So for example, and here's a little bit of a hint, do you know if a lactobacilli, and you don't even have to know this, you can just think about this, is lactobacillus an anaerobic or an aerobic bacterium? But you can actually answer this just by knowing what you know right now, right? Oh, everyone, complete silence. This is amazing. So in other words, where is it anaerobic? Your gut is anaerobic. Is lactobacillus only found in your gut? Something to think about a little bit maybe. And those are the types of integrative questions. As long as you've studied the material and have had time to think about it, it won't be any issue for you. By the way, lactobacillus is an anaerobic bacteria. We find it on our skin. We find it as an early colonizer, and it disappears with time, right? And all of you should know that because it's in your notes. And if you've gone through it, you know that it's clearly outlined for you in your notes. So is there anything else that I wanted to tell you? This will also be something that you should be thinking about. You won't get the whole question, but this will also appear for you on your midterm as well. And this is another example of the types of questions that I like to have. So if you've accidentally changed the number of gut epithelial cells, what sorts of things might you expect to see? Which begs the question, what do gut epithelial cells normally produce? So this goes back to a couple of slides. There are actually two slides that deal with gut epithelial cells, the ones that are found in the mucosal lining. If you lose those gut epithelial cells, what might you expect to see? Will you see an increase in some things? Maybe. Will you see a decrease in the expression of some things? Maybe. And these are the examples of some of the more challenging integrative questions. As long as you studied, as long as you've gone over your notes, there's not going to be anything really crazy difficult about it, but it's not something where I'm just going to ask you which of the following bacteria are found on your skin. You'll get some questions like that, to be honest, but you'll also get some questions like this. Do you have any questions about the questions? No? So I am around all of next week. I'll be in the office every single day next week. You're free to drop by. You're free to come in for breakfast with me if you want. You're free to eat Twix if you want in the office. I have lots of Twix in the office as well. Good luck on your studying, and I'll see you in class on Monday. Not next Monday, the week after.